my name is Beverly Greenwood van Meerveld, and I'm the immediate past president of the American Neurogastroenterology and Motility Society and the current chair of the ANMS Institute. I'd like to welcome you today to the ANMS virtual symposium entitled Mechanisms of Normal and Abnormal Upper Gut Motility. Now, the goal of these virtual symposia is to keep you up to date with the latest research in the field of neurogastroenterology motility until the time comes when we can meet once again in person at our annual meeting. And I'd like to remind you that the ANMS 2021 meeting will be held in person in Boston on August 13th to August the 15th. We have a record number of abstracts submitted to the meeting, and we look forward to seeing you in person at that meeting. It's now my pleasure and honor to introduce today's session moderator, Dr. Gary Mawe. Dr. Gary Mawe is the Samuel W. Thayer Professor of Neurological Sciences within the College of Medicine at the University of Vermont. Dr. Mawe has been a long-standing member of the ANMS and has also served on the ANMS Leadership Council. Most recently, he and his colleagues have been investigating the pathophysiological changes in mucosal serotonin signaling, as well as enteric neuronal excitability and neuromuscular transmission that contributes to altered gut function in colitis, as well as functional GI disorders and multiple sclerosis. Thank you so much, Dr. Mawe, for agreeing to moderate this ANMS-sponsored virtual session. And we're really looking forward to hearing the presentations. Thank you.
Thank you, Beverly. And thank all of you I want to thank all of you for attending today's session. It's a beautiful day outside in most of the US, and I'm glad you're here. We have three outstanding speakers in today's session, and they'll be introduced individually as we go along. Any questions that you have, please enter them in the Q&A box that you'll find at the bottom of the screen and indicate which speaker you would like to have address that question. Or you can indicate that you'd like the panel to address the question. They'll be answered after all three talks are completed. So we'll be having a Q&A session at the end of this hour. Any questions that we don't get to during this hour will be answered on the Doc Matters website. So let's get started here. Our first speaker is Dr. Prakash Gyawali, who's Professor of Medicine, Director of Neurogastroenterology and Motility, and Program Director of the Gastroenterology Fellowship Training Program in the Division of Gastroenterology at Washington University School of Medicine. His academic interests include GI motility, GERD, and functional disorders. He directs the GI motility centers affiliated with WashU, and he's involved in motility testing using high-resolution manometry, ambulatory esophageal pH and impedance monitoring, and also wireless pH monitoring and EndoFlip. Today, he'll be giving a talk entitled Esophageal Motility and Dysmotility, Physiology and Pathophysiology. Welcome, Dr. Gyawali.
Thank you, Gary, and thank you to the ANMS for asking me to give this talk. I'm going to share my slides here. My disclosures are at the bottom of this slide. I'm going to try and touch on motor physiology and pathophysiology from an obstructive standpoint, obstructive motor physiology and pathophysiology, and then move on and talk about the exact opposite, pathophysiology of GERD. And then finally, I'll talk a little bit about esophageal sensation if there is time and try to tie everything in together. So let's talk about the esophagus in general. There is skeletal muscle at the top of the esophagus, the proximal third of the tubular esophagus, and the remainder is smooth muscle, an inner circular layer and an outer longitudinal layer. There is a sphincter at either end, the upper esophageal sphincter, which is skeletal muscle, and the lower esophageal sphincter, which is smooth muscle. Now, brainstem nuclei directly innervate the skeletal muscle portions of the esophagus, the sphincter as well as the proximal third. And so this portion has direct innervation from the lower cranial nerves. The remainder of the esophagus is innervated through these intermediary ganglia in the intramuscular orobox plexus. And through these ganglia, we have excitatory innervation to the longitudinal muscle and to the circular muscle. And we have inhibitory innervation to the circular muscle. And that inhibition is really important. We'll discuss this in just a minute. The lower esophageal sphincter is controlled by its intrinsic tone through calcium-related mechanisms and by both vagal stimulation and vagal inhibition. And in particular, vagal inhibition is necessary for sphincter relaxation. Now, the gradient of neuromodulators in these nerves that directly control the smooth muscle determine whether there is excitation or inhibition as the dominant phenomenon. So in the proximal esophagus, in the proximal of those two smooth muscle segments in the esophagus, acetylcholine is the dominant neuromodulator, and that's an excitatory neuromodulator. In the distal esophagus, there is more of the nitric oxide-based neuromodulator, and that is more of an inhibitory neuromodulator. And a balance between excitation and inhibition determines this nice peristaltic antegrade movement of circular muscle contraction in the esophagus. Now, if inhibition were to be blocked, you could get a more exaggerated contraction because you need inhibition to dampen the contraction, and you'd get a more vertical contraction, a more simultaneous or premature contraction. So inhibitory dysfunction is what determines these more spastic, more exaggerated contractions and the more premature type contractions. How was this determined? Well, this was first described by Daniel Sifrin because you cannot actually see the inhibition when you do motility testing. He developed an ingenious method by using a balloon with a sensor tied to a motility catheter. With the balloon deflated, you see the contraction sequence going from top to bottom. With the balloon inflated, you can see a decline in the measurement through the sensor in the balloon, which determines, or which is actually a measure of inhibition prior to contraction. Let's see this in a high resolution manometry Klaus plot. This is a Klaus plot anchored by the upper esophageal sphincter, band of pressure at the top, lower esophageal sphincter, band of pressure at the bottom, and the peristaltic sequence going from top to bottom. Excitation determines contraction of the smooth muscle. Inhibition times the contraction. So sequencing and dampening of the contraction so that the top part squeezes first and the bottom part last. And like I said, inhibition is important for relaxation of the lower esophageal sphincter. Now, what would happen if inhibition were abnormal? If you look at panels B and C, contraction wave abnormalities. These are situations where with the standard line tracing manometry, you see exaggerated double-peaked, multiple-peaked contractions. You can see that the dip in the second half of the panel, the yellowed out portion, is less than what you see with normal contraction, meaning that inhibition is abnormal. When inhibition is abnormal, contraction can be exaggerated. The bottom panel, distal esophageal spasm, you can see that the contraction is more vertical from top to bottom, and again, inhibition is abnormal. Let's see this in a high resolution manometry contraction. So if inhibition were to be abnormal, then sequencing would be abnormal and dampening of contraction would be abnormal. So you could see an exaggerated contraction and a premature contraction. So this is what would happen in the esophageal body. If inhibition were to be abnormal, you would probably also have abnormal lower esophageal sphincter relaxation. Why is that important? Well, that is the prime mechanism that leads to esophageal outflow obstruction in achalasia, where a normal looking esophagus turns into one of these dilated esophagi with a tapered bird's beak appearance at the bottom. So the abnormal esophageal function here relates to abnormal LES relaxation, abnormal body peristalsis. This happens because of loss of inhibitory nitric oxide mediated innervation through gradual loss of the myenteric plexus ganglion cells through an immune mediated inflammation. The incidence is about 2 to 3 in 100,000 population. These are the cardinal symptoms: dysphagia, regurgitation, weight loss, and chest pain. And these are some of the complications: aspiration pneumonia, squamous cancer, and a higher mortality. Now, if you take esophageal outflow obstruction, achalasia is not the only cause. You have structural or mechanical strictures, other abnormalities at the esophagogastric junction, and certain motility type processes. Opioid effect has been recognized as one of the mechanisms for outflow obstruction, a reversible element. But if you were to identify abnormal relaxation of the lower esophageal sphincter, there are several mechanisms. Idiopathic, infectious, neoplastic are some of them. The idiopathics can be of two types: genetic predisposition, that is achalasia, where through an environmental trigger, you have neuronal inflammation or neuronal death. If there is neuronal death, you get no body contraction, type 1 and type 2 achalasia. Everybody has abnormal lower esophageal sphincter relaxation here. Neuronal inflammation, the nerves are not dead. You get an imbalance between excitation and contraction. So you may have the premature or exaggerated or intact contraction. That's where you have type 3 achalasias, and EGJ outflow obstruction with intact esophageal body peristalsis. Recently, we have recognized that an eosinophilic pattern can also result in a similar end phenotype with abnormal LES relaxation and abnormal motility. Stu Speckler has done a lot of work with this. An eosinophilic mechanism, infiltration of the muscle of the esophagus, can result in an achalasia-like pattern. Now, the infectious type, Chagas disease, we all learn about this in medical school. The pathogenesis is unclear, but there is a bug that is associated with it, the Sese bug, and the parasite is Trypanosoma cruzi. Now, we also know pseudoachalasia from neuronal death or inflammation, and this can resemble achalasia. You make a diagnosis based on the clinical scenario. Let's stay with the lower esophageal sphincter. The dysfunction of the sphincter can be at the opposite end of the spectrum. You can have excessive relaxation or inappropriate sphincter relaxation, transient LES relaxation, and that is one of the prime mechanisms of gastroesophageal reflux. You can also have disruption of the esophagogastric junction, and that's in the form of a hiatus hernia. And what refluxes may not be adequately cleared if there is a hypomotility disorder in the esophagus. There are a couple of downstream factors: delayed gastric emptying, increased acid production, and upstream factors: xerostomia, meaning lack of saliva, impaired swallow initiation. So both hypomotility mechanisms and structural mechanisms can participate, and indeed, a hiatus hernia can worsen or make the motility abnormality at the EGJ, esophagogastric junction, or the esophageal body more profound. Now, all of these can lead to elevated esophageal acid exposure time, and that defines gastroesophageal reflux. Now, the esophagus is very unique. It has both primary peristalsis that we initiate in the skeletal muscle esophagus volitionally, and secondary peristalsis. Now, this is a nice study from a little while ago now. Anesthetized cats with esophageal perfusion. N is no fluid, no perfusion. PBS is phosphate buffered saline, and HCl is hydrochloric acid. So if there was volume in the esophagus, that initiated secondary peristalsis. But if you put acid, you actually initiate primary peristalsis. And this is by the acid resulting in saliva secretion through an esophagovagal reflex. And that saliva secretion generates primary peristalsis. Indeed, the volume of saliva secreted is higher when there is acid. So the esophagus is able to discern the presence of acid. Secondary peristalsis is triggered by distension. Primary peristalsis triggered by acidification of the esophagus. The brainstem is involved, and there are vagal activities associated with that. How is this important? This is a nice study where radio-labeled acid was instilled into the esophagus. And you can see the esophageal pH drop when that happened, but the volume cleared very quickly, but in a stepwise manner. And each step was associated with a DS, a dry swallow. That is actually a swallow that the patient initiates because of saliva secretion. If you were to suck the saliva out using one of those little suction things that you use, that the dentist uses, you can see that the esophageal pH, the second line graph there, never corrects despite the dry swallow. So secondary peristalsis and primary peristalsis are important. So this is an actual pH impedance tracing, a reflux episode. The volume is cleared by a peristaltic sequence that is either primary or secondary, but within 30 seconds, in many situations, you have a post-reflux swallow-induced peristaltic wave that brings saliva to the esophagus to clear mucosal acidification. And the frequency of PSPW can actually be measured. Patients with refractory reflux disease tend to have a lesser degree, a lesser proportion of PSPW. Let's talk about a couple of other interesting things about the esophagus. So if you infuse air into the esophagus in the upright position, that causes the upper sphincter to relax, so you belch it out. If you infuse liquid in the upright position, the upper sphincter contracts, the LES relaxes, and there is secondary peristalsis. The esophagus is able to discern air versus liquid. If you do this in the supine position, again, the upper sphincter contracts, there is secondary peristalsis, and the lower esophageal sphincter relax to push the refluxate or the content in the esophagus into the stomach. Now let's talk for a second about sensation. I guess I have a couple of minutes left. Underlying motor disorder can be associated with sensation. And indeed, the perceptive thresholds and the pain thresholds in the esophagus vary by esophageal phenotype. In functional heartburn, the threshold is the lowest, meaning the patient has the highest perception of esophageal sensation. And you can see that the threshold is still lower in non-erosive reflux disease and erosive esophagitis. Indeed, acid infusion into the esophagus in these barostat-based studies or electrical stimulation-based studies, acid infusion reduces both the perception threshold and the pain threshold. And in predisposed individuals, patients who are predisposed to have esophageal visceral hypersensitivity, they have lower pain threshold drops than healthy individuals. And indeed, if you treat these patients with PPIs, the pain thresholds may improve, but never return to normal. And the hypersensitivity to acid persists on repeated challenge. So in other words, people who have a pattern of esophageal hypersensitivity can redevelop the situation, the symptoms, if they are re-exposed, if acid is a trigger. Now, let's talk for a second about FLIP. This is the precursor of FLIP. This is an experiment done by Satish Rao in 1996. We're talking about FLIP now in a very different format, but impedance planimetry used to measure cross-sectional area. And Satish Rao demonstrated that in patients who had these spastic disorders and chest pain, they had a reactive esophagus, and that reactivity was more pronounced, meaning when he distended the balloon, the esophagus tended to squeeze or contract a little harder to try and move the balloon out of the way. And indeed, the higher the contraction amplitude, the lower the pain sensitivity. So patients with high contraction amplitudes tend to have more pain or more esophageal hypersensitivity. So ladies and gentlemen, motility and sensation are related. And so aging, decreased motility, decreased sensation. As you go down this list, Barrett's esophagus, decreased motility, decreased sensation. Connective tissue disease, same thing. But stress experiments, functional symptoms, the motility is exaggerated. These are who have high amplitude contractions. They also have higher perception and more pain. Achalasia in early stages, you have exaggerated contraction, the type 3 achalasia, increased perception, more chest pain. But in late stages, type 1, type 2 achalasia, there is hypomotility of the esophageal body and decreased esophageal perception. Thank you very much. And I look forward to your questions at the end of the session.
Thank you, Prakash. That was a fabulous talk. Our next speaker is Dr. Shanthi Srinivasan, who is professor of medicine and director of the Division of Digestive Disease at Emory University. Her research interest focuses on the enteric nervous system and pathophysiological mechanisms underlying GI motility disorders. She also studies the role of neurotrophic factors on regulating hepatic steatosis. She was recently awarded the VA Senior Clinician Investigator Scientist Award. She's a counselor for the American Neurogastroenterology and Motility Society, and as of July 1st, she'll be the chair of the editorial board for Neurogastroenterology and Motility. Her talk is titled Cellular Mechanisms Underlying Gastroparesis. Welcome, Shanthi.
Thank you, Gary, for that really nice introduction. And I also want to thank the ANMS for giving me this opportunity to talk. I'm just going to take a minute to share my slides. Sure. So my talk today is on cellular mechanisms underlying gastroparesis. I have no disclosures. So as we all know, gastroparesis is associated with delayed gastric emptying without, mechanical obstruction. There are a very important component of gastroparesis is the symptoms associated with it. So it's not necessary that, delayed gastric emptying has to be a part of the sort of the definition. And as we know, we are all going by the Rome 4 criteria. And we all know that it's associated with nausea, vomiting, early satiety, belching, bloating, and upper abdominal pain. So focusing on the various aspects of the stomach, we all know that the fundus, the accommodation in the fundus, that plays a very important role in terms of symptomatology in gastroparesis. The body, in terms of its contractile activity, Prakash very nicely explained the smooth muscle contraction and the importance of excitatory and inhibitory neurons. So the body contractility is a very important component in not only grinding of the food, but ultimately gastric emptying also. Then we have the antrum, where again, there's a lot of grinding and mixing. And then finally, the pylorus involved with, the resistance, and then ultimately allowing the contents of the stomach to empty into the duodenum. So all of these various aspects of the stomach, play an important role in the pathophysiology of gastroparesis. And in this very nice review recently published by Mike Camilleri, he highlights the importance of looking at the various aspects of the stomach and the relationship of motor mechanisms to gastroparesis symptoms. And he talks about the importance of accommodation and how, if modulating modulating the neurotransmission from the stomach to the brain. And I think we're going to hear more about this in the next talk. So this was a nice review, and I wanted to highlight that the motor mechanisms relating to gastroparesis symptoms are very important and can actually lead to individualized treatment. So when we think about the pathophysiology, as I just mentioned, it's not only the smooth muscle, but the various components, the autonomic nervous system, the enteric nervous system, the interstitial cells of Cajal, and how ultimately they affect gastric accommodation, the antral hypomotility, and pylorospasm can all contribute to the pathophysiology of gastroparesis, and in addition, small bowel motility. I just have one slide talking about potential etiologies of gastroparesis. As we all know, a lot of the most common causes of gastroparesis are seen in patients with diabetes or post-surgical complications leading to gastroparesis, and then, of course, the idiopathic gastroparesis. Other diseases that can be associated with gastroparesis are neurological diseases such as Parkinson's or paraneoplastic syndromes, metabolic disorders like hypothyroidism, medications, narcotics, anticholinergics, in rheumatological disorders, such as scleroderma and myopathies. We can see delayed gastric emptying and gastroparesis, psychiatric disorders, eating disorders, and finally diffuse GI disorders also. So in all of these different, with all of these different etiologies, what could be affected? One of the things I mentioned is gastric accommodation. And as was nicely explained in the previous talk, a balance between the inhibitory component and the cholinergic component can actually result in it that is required for normal gastric accommodation. And if there is that imbalance, that is affected and can lead to the symptoms of gastroparesis. What I wanted to do in this talk, as I was assigned to talk about cellular mechanisms, I'm going to highlight three aspects, three recently published papers where these different groups across the country look at cellular mechanisms of gastroparesis. One group that I will talk about right now is from the Mayo Clinic with Dr. Adil Barucha and Dr. Tamas Ordog. They recently published this article in JCI Insight, and this highlights the role of mitochondrial gene expression contributing to diabetic gastroenteropathy. And I wanted to spend just a few minutes on this paper. So what Dr. Barucha and his group did was they looked at duodenal mucosal mitochondrial gene expression. What they found was, so basically they had a nice cohort of patients with diabetic gastroparesis, and they compared these with control subjects, patients that did not meet the criteria for gastroparesis. They were able to obtain duodenal mucosal biopsies, and they looked at entire gene expression, nuclear mitochondrial gene expression, and they focused on oxidative phosphorylation genes. And what they found was genes related to oxidative phosphorylation were affected in the patients with diabetic gastroparesis or diabetic gastroenteropathy. And this was very nicely demonstrated. They found a nice correlation. There was a reduction in the mitochondrial gene expression, particularly the ones related to oxidative phosphorylation. So one of the conclusions they had was that the changes in mitochondria can contribute to changes in smooth muscle, as well as in neuronal changes, ultimately leading to diabetic gastroenteropathy. So this is one cellular mechanism I wanted to highlight. The other group that I wanted to talk about is the recent publication related to the role of immune profiling in gastroparesis. This is the group from Stanford with Ida Habtezian and the first author, Andres Gottfried Blackmore. And what this group did was they looked at gastric mucosal immune profiling and wanted to see that in a subset of patients with idiopathic gastroparesis, whether they could phenotype and find changes in comparing to normal controls, whether there was a specific upregulation of a specific type of, let's say, macrophages or T cells. And as you see here, what they found was in gastroparetic, they pictorially depict here that in patients with gastroparesis, and they defined it both based on symptoms as well as delayed gastric emptying, there was an increase in mucosal macrophages as well as CD8 positive T cells. And then they went on to do a lot of correlative studies and phenotyping, and also they used a specific a novel method called CyTOF to do this large scale phenotyping. What was different about this group was, I think a lot of work has been done on the muscularis macrophages and immune cells. So here they were looking at the mucosal macrophages, and their hope is that ultimately, if they can further, take these studies further, this can be used as a way to sort of as a window to maybe identify or define or ultimately even, treat gastroparesis, and they focused on idiopathic gastroparesis. So this was another a recent paper where they talked about potential cellular mechanisms for gastroparesis. And the final paper that I want to highlight is actually done, work done by the Pandu Gangula group. They are at Emory University and Vanderbilt University. And they have been doing some work on nNOS signaling and the role of antioxidants and nerve. We've been fortunate to participate with them. And as one of their articles that they published last year showed the role of GSK3 beta, and using a high-fat diet model, they tested gastric emptying. And what they found was that gastric emptying was delayed in a high-fat diet model, and this could be reversed with GSK3 beta inhibitors. This group studies a lot on, nNOS signaling, how it's regulated by NRF2 and antioxidants. And as is shown in their next paper, they had an important observation where there was sex differences, and they showed the role of sex hormones in regulating, gastric NRF2, so the antioxidant function, and they're using an experimental model of hyperglycemia. So I think antioxidants play an important role in regulating gastric emptying, and I think the focus here is on the neuronal NOS and also survival of nitrogen neurons. So today, my goal was to show you to show you these studies where recent studies where they've looked at immune mechanisms in gastroparesis, the muscularis mucosa, immune cells within the mucosa versus the muscularis mucosa, the role of mitochondrial dysfunction, and how studies to phenotype, oxidative phosphorylation and genes related to that in diabetic gastroparesis. And finally, the importance of gender differences, the role of NRF2 and the nNOS pathway affecting neurons, and and thereby affecting gastric emptying and the gastroparesis. What I've not had a chance today, in the interest of time, to talk about the interstitial cells of Cajal, some recent smooth muscle papers where they've looked at changes in smooth muscles, the glial cells, gut microbiome, and interactions with ENS. And hopefully at a later time, we can discuss this. So I look forward to your questions, and thank you for this opportunity.
Thank you, Shanthi. Once again, for those of you in the audience, any questions that you might have, please enter them in the Q&A box that you'll find at the bottom of the screen. Our next speaker is Dr. David Levinthal, who's an assistant professor in the Division of Gastroenterology, Hepatology, and Nutrition at the University of Pittsburgh School of Medicine. And he serves as the Director of Neurogastroenterology and Motility Center at Pitt Medical Center. He also co-directs the Program for Gut-Brain Health, an integrative behavioral gastroenterology clinic for patients with functional GI and motility disorders. He leads an active research program spanning clinical, translational, and basic science approaches aimed at understanding the neural basis of mind-body interactions and their role in determining states of GI health and disease. Thanks for joining us, David.
Okay. Well, thank you very much for that introduction, Gary. I'm just going to share my screen here and get this talk started. So I'm really happy to be invited to participate today. Hope everyone can see my screen. So I'm going to talk about some of our recent basic science work that bears on motility, focused on the brain-gut axis. and some of our recent findings about how the cerebral cortex can bear on motility, specifically focusing on the stomach. So I have no relevant disclosures. So the brain-stomach connection has been a part of the story for gastroenterology from the beginning. I think it's been widely appreciated for well over 100 years, probably well over 150 years or more, that how we think, how we feel, how we move, all have influences on gut function. These were some of the first observations that were the basis for GI physiology, from William Beaumont in the 1830s to Ivan Pavlov's work in the late 1800s to the current day. And in general, and this is a broad statement, of course, but neural influences on the stomach, as we heard about from Prakash's talk, the parasympathetic innervation is really important for motility. We know that neural influences on the stomach really come from two different inputs, the parasympathetic nervous system and the sympathetic nervous system. And in general, they're not complete opposites, they work in concert, but in general, parasympathetic activation increases gastric secretion and promotes meal-induced motility patterns. Sympathetic activation tends to decrease gastric secretion and inhibit some of those patterns. That's a broad statement, but in general, that holds. And it goes without saying that the autonomic regulation of the stomach itself shapes the gastric microenvironment in ways that probably do influence the microbiome. So we don't talk enough about top-down influences on the microbiome, autonomic influences, but there easily could be. So the question becomes, we know that there's an autonomic nervous system control of almost every organ in our body, including the GI tract. And the classical view has been that this autonomic regulation happens in some homeostatic way, that there's a set point and peripheral feedback that programs the balance of these outputs and control of the organ function. And that extreme behavioral states may shunt regulation in one way or another. For example, a fight or flight response, driving sympathetic activation, kind of external action focused, or that parasympathetic activation in a rest and digest or internal action focused behavioral state shapes autonomic output. But we know a little bit more now about autonomic function, and there's an increasing appreciation that allostasis is this concept that autonomic function really is a top-down model, that there's a predictive quality to our autonomic control. And it's much more nuanced than just a feedback and a homeostatic set point. And if you think about allostasis in a brain controlling our autonomic output, that really can account for how mental events, emotion, behavioral planning really play out with autonomic activity changes. So the question becomes, how does the cerebral cortex influence the autonomic nervous system? So that's going to be the focus of my talk today. So to answer this question, this is actually a really basic question about the nature, the neurobiologic nature of the brain-stomach connection. There's no one neuron that goes from the cerebral cortex to the stomach. It's a neural circuit. How could you trace a neural circuit? So it turns out that rabies virus, actual rabies virus, is a neurotropic virus that is ideally suited to answer questions of neural circuit function and connectivity. So it's an ideal viral neural circuit tracer. Rabies virus has a very unique property that it infects neurons that are near the virus. The neurons take up that virus, retrogradely transport it to its neural cell body. This would be a number 1 neuron in the schematic on the right. And that virus replicates and then passes the virus onto all of the inputs to that infected neuron. So transsynaptically crossing a synapse to a second neuron that has inputs to that original infected neuron. The virus then replicates again and passes again transneuronally and so on. So through a neural circuit of a chain of connected neurons, rabies virus can reveal a neural circuit of multiple neurons, 3, 4, or more. It's a time-dependent process. It works in all mammals. And basically, this is the technique that we've used to reveal the cerebral cortex control of the stomach. So the nature of this experiment that we just recently published last year in PNAS, basically, this is the schematic model of the experiment. And I think it really highlights this fundamental question about what regulates parasympathetic output or sympathetic output to the stomach. So the basics of the experiment are that we injected the stomach of rats with rabies virus. And all the neurons that were located in the enteric nervous system and the extrinsic inputs to the stomach got infected with rabies virus, took the virus up to their cell bodies. On the left-hand panel in panel A, you can see that this went through the parasympathetic network, infecting the dorsal motor nucleus of the vagus nerve, replicating, passing its inputs to the nucleus tractus solitarius, and then replicating and then infecting inputs to those cells, which gets you to the very closest linkage between the cerebral cortex to the stomach, which would be cortical layers, cortical neurons in layer 5, the output layer of the cortex. That would be a 3-neuron system. On the right-hand column here in the diagram, panel B, we were able to restrict the progress of rabies virus to only the sympathetic nervous system by cutting the vagus nerves below the diaphragm prior to our injection into the stomach. This allowed the virus to propagate through sympathetic channels. And there's an additional neuron to get to the cerebral cortex, requiring an additional survival time, extended survival time, but nonetheless infecting layer 5 neurons, output neurons in the cortex that were linked to the sympathetic control. So I hope this experiment looks clear to everyone. Panel A versus B, we can distinguish between the cortical output that controls the vagal output to the stomach from the sympathetic output. So this is a picture of some of the neurons that we were able to label with a rabies virus antibody and reveal the location in the cortex. Shown in the panel on the right here in green are neurons that are infected with rabies virus after injection into the stomach. These are neurons that happen to be located in the insular cortex for this graph, this picture. And these neurons were localized only to layer 5, the cortical output layer of the cortex. So I'm going to show you now a map. And I don't want to spend too much time on the neuroanatomy here, but all I need everyone in the audience here to appreciate is that in panel A on the right here and panel B are 2 maps of the rat's cortex. And as you can see, the parasympathetic map of where neurons are located, infected neurons from injection into the stomach, there is a red circle here around a area what's in the insular cortex. And then on the right, you can see a cluster of neurons that are located in a different area. And this happens to be a motor area. It's very clear that by looking at these 2 maps, that the cerebral cortical output to the stomach of the 2 different arms of the autonomic nervous system originate from 2 very distinct cortical networks, which I think is a really important insight. If you look in a textbook about what areas of the cortex influence the stomach, there's a lot of discussion about the medial prefrontal cortical area. We saw cells there. But it doesn't seem that that is the primary source of the output to the stomach. It's really the insular cortex for parasympathetic control and motor cortical areas for sympathetic control. So I'm going to go into a little bit of detail about this, not too much, but I want to talk about the implications of this basic observation. So the first major finding was that the rostral portions of the insular cortex form essentially most of the output for the parasympathetic control of the stomach. Over 85% of the neurons that we found in the maps of injected stomachs that went up the vagus nerve originated in that location. It was a bit bilateral. If you blink too fast, you would have missed it, but I didn't talk about bilaterality, but it's both cortexes, a little bit more on the right than the left. But the rostral insular cortex contains all of those neurons. And it's fascinating to really think about how close that is to the control of the stomach. This is a 3-neuron system: rostral insular cortex, a relay in the brainstem, and an output from the vagal nuclei directly to the stomach. That network architecture, a 3-neuron system, is identical to what controls even the skeletal motor system in a rat. Humans and some non-human primates have a 2-neuron motor system, but most of the neural output to control our skeletal motor system is a 3-neuron system. So in many ways, the rostral insular cortex can be viewed as the parasympathetic motor cortex. And I think that's an important finding, because now we really have a handle on what area of the brain should have the most influence on stomach output. Okay. The other implication of finding that the rostral insular cortex forms the major output to the stomach, parasympathetic control of the stomach, is that this area is generally regarded as a visceral sensory area, receiving inputs from from the stomach and other GI tract organs, and actually other organs as well, heart and so on. So we found that a visceral sensory area is actually the source of visceral output. And that kind of creates a loop, which is actually kind of fascinating, that there must be some visceral sensory and motor integration that happens in this cortical site. And in a way, because this site is so important for regulating the stomach, other cortical areas that input to that area must have an influence as well. So this kind of brings in the idea that what we think and how we feel and what we expect to happen play out with changes in our stomach function, and even interpreting the signals that efferences, or afferences from the stomach to the brain. So there's really a loop, and I think that's another important insight. So I want to just touch on one other point, and then I think that'll be the end here of this. But I just want to talk about the sympathetic map. So again, this panel to the right, we found when we cut the vagus nerves and let the virus go up, the sympathetic output circuit, a totally other brain area was labeled. And this brain area, and there's just too much to go into the details here, but all I want you to know from this diagram is that the motor cortex area that was labeled, just like there's a homunculus in the human brain, there's a ratunculus in the rat brain. And what I've labeled here is kind of the motor areas that are controlled by various sites in the motor cortex. The cells that we labeled from this experiment are in blue, and a former experiment we published from the kidney are labeled in yellow. And as you can see, in both a kidney map and a stomach map, the sympathetic control of those organs seem to be embedded very heavily in the trunk control areas of the rat's motor cortex. Over 95% of the neurons were located in these regions. And I think that's an important insight as well, because interestingly, the thoracic muscles, our abdomen, seem to be the same area of the brain that control organs that are located in that anatomic area. And it brings up the idea that how we use our trunk muscles could actually influence organ function. So maybe it's not too outlandish to think that yoga, Tai Chi, or Pilates, or any kind of postural focused motor program could have an influence on how our body works, or even our abdominal wall tone. So I'll leave it there. So basically what we've shown is that there are two cortical networks that seem to regulate both the parasympathetic and sympathetic output to a single organ, the stomach. And that these two arms of the autonomic nervous system subserve many different disparate functions and are pushed in a fight or flight or rest and digest behavioral state. And they originate from very distinct cortical networks that seem to be linked to those behaviors. Fight or flight being linked to external action or movement, rest and digest linked to kind of an internal or emotion or internal mental state function. And it's fascinating that this really plays out with the neuroanatomy. So could there be a link between these circuits and their function with programming stomach function? I would wager that this is important for programming stomach motility, and it could influence the microbiome by shaping the gut microenvironment. So what I hope this work will become the seed for, and this is my goal research-wise, is not just to learn about rats, but to also do a parallel experiment in monkeys, and we're doing that right now. I'll tell you that the rostral insular cortex story holds in monkeys as well, and that all of the sites I just showed you in these maps become cortical targets for potentially using brain-based therapies for influence in the stomach. So I want to thank everyone for their attention, and thank you for the invitation. I'm happy to do questions, and I think we have enough time. So I'm going to stop my screen. Thank you.
Thank you very much, David. That was a great talk. I enjoyed all three of them. Anybody who has questions, please enter them in the Q&A box. At this point, we only have one question, and that is related to the esophagus. Is eosinophilia an association with achalasia due to food stasis or cause? Prakash, could you address that one, please?
Yeah, that's a very good question, because right now, eosinophilia in the context of achalasia only has hypotheses and not real answers. And stasis is one of the hypotheses, but it's not one of the leading hypotheses, because if you were to relieve stasis, you do not relieve eosinophilia. In other words, if you took a patient with eosinophilia in the context of achalasia. And a disruption of the lower esophageal sphincter, a myotomy or a pneumatic dilation, and rechecked for eosinophilia, it does not consistently go away, which means that stasis is probably not the primary mechanism. The other hypotheses linking eosinophilia to achalasia is that the neurotoxins, some of the products of the eosinophil, may be neurotoxic and may result in a partial or complete loss of neuronal function. Again, it's not a very solid hypothesis, because if that were to be the case, if you treated the patient with steroids, function should return. So in other words, if you got rid of the eosinophil, just like with EOE, some of that function should return, and it doesn't necessarily return. So I think the bottom line here is that the pathophysiology of eosinophilia and achalasia is complex. There's probably more than one mechanism, and there is probably a point of no return beyond which the damage to the neuronal mechanisms is more permanent, or the damage that subsequently ensues in the muscular mechanism is more permanent. We don't completely understand that. Another issue is that the manifestations of achalasia. So when we study patients with achalasia, when we have access to these patients with achalasia, they are symptomatic, and they are a few years down the road from when the initial triggering mechanism happened, and that is a problem as well.
Okay, thank you. The next question is for David Levinthal. Electrical neuromodulation, such as auricular neurostimulation, seems to help patients with functional GI disorders. Does it matter to do the treatment on the left or right side, considering the nerves and the ANS pathway?
Okay, that's a great question. So I want to highlight, I mentioned laterality briefly when I spoke. So just to be clear, the way we did our experiment was to inject the anterior wall of the stomach alone. So I didn't inject the entire stomach. And it's interesting that might have some preference for left vagal innervation. To some degree, it's not a balance of where the vagus nerve hits the stomach. So if one can regard the laterality of our injection as being left vagus, there seemed to be some bilateral representation, more on the right, contralateral, if you will. I suspect that both sides would be needed, because you have an entire stomach, it's not just one single wall. And I think to be kind of clear, I think both ears for auricular stimulation probably are needed. But I think there's bilateral representation for the vagal control, heavily skewed contralateral, if you will, side of the stomach, or the right side of the cortex for the sympathetic. So that might be a different story, but auricular might be bilateral.
Thanks, David. Okay, let's jump to a question for Dr. Srinivasan from Henry Parkman. How is the duodenum influencing gastric hypomotility of the stomach? Are there mitochondrial dysfunctions also in the stomach?
So that's an excellent question. So I think and probably you're referring to the study that was published in JCI Insight, where they used duodenal mucosal biopsies. I think they used that sort of as a snapshot to assess changes in mitochondrial function. Feeling similar changes would be present in the gastric mucosa also, and then ultimately also in the sort of smooth muscle and neuronal mitochondrial dysfunction may also be, playing a role. So I think this is an initial study that they hope to extend to better understand mitochondrial function in both in the smooth muscle as well as the neurons, ultimately, playing in terms of the gastric contractility. So yeah, I think these are in the beginning stages, and then we need to investigate and understand it further. Thank you.
Okay, so here's a question for, was somebody going to add something to that? Okay, this question, any panelist can jump in on. What do you think is the mechanism of gastroparesis in patients with Roux-en-Y gastric bypass, and what kind of treatment would best help them?
My answer to that would be, how did we determine that there was gastroparesis when the stomach was bypassed, and did it pre-exist? So, the emptying out of the pouch is different. It's emptying by pressure. It's not really by peristalsis, because it's a mechanical emptying. So after a bypass is placed, it's very, very difficult to determine what's happening with the native stomach. And so I would be curious as to how the person asking the question determined delay in the native stomach following gastric bypass. I don't know if David or Shanthi have any comments, but this is from outside looking in for me.
I mean, I think one has to just kind of, just thinking through the question, how new is the surgery, and is it local swelling of the anastomosis and all the problems with kind of more mechanical blockage rather than a motor dysfunction of the stomach. I agree with you, Prakash. I mean, it's basically a drainage model at that point. So pressures matter so much, and the angle of that connection and so on. So hard to answer.
Okay. David, could you give potential examples of therapies for the CNS to control gastric function? Is this brain stimulation or pharmacologic, would you think?
Oh boy, I have to give away the whole story, huh? So that's what I'm going to try to do for the field, right? So, all of the areas that I mentioned, in these experiments, create a map, right? And we don't know what these areas do functionally. There will be many ways to stimulate the brain non-invasively. You could use transcranial direct stimulation, a direct current stimulation. There's transcranial magnetic stimulation. That's something I'm interested in. I have a lab, we're starting to do that. So, the whole basis of these maps is to kind of create a logic for seeing what happens to the stomach when we stimulate. So I could imagine a future where some technique could focus stimulation, neuromodulation, to alter the sensitivity or the long-term plasticity of a cortical area. That's used for treatment for depression. And, we just need a better target, right? So, so the FDA has approved TMS for depression. One could imagine a future where we do that for a functional GI disorder.
Thanks, David. Prakash, do you think longitudinal muscles play a role in esophageal peristalsis and esophageal symptoms?
Yes, they do. And there's been a lot of work done through primarily Ravi Mittal's lab in looking at longitudinal muscles and their function. In normal emptying, yes, you need the longitudinal muscles to lift up the esophageal gastric junction for effective emptying from the esophagus. And the longitudinal muscles also play a role in a phenomenon known as pseudo-LES relaxation. You see these longitudinal muscles pull up the esophagus into the thorax when there is no opposition at the bottom. And so the LES may move away from the recording sensors at the LES level and fail to demonstrate abnormal LES relaxation unless you follow the LES up into the chest, and high resolution manometry allows you to do that. Now, longitudinal muscles are also important in how the different subtypes of achalasia behave. And Ravi has shown that the intactness of longitudinal muscles probably helps type 2A patients behave different the subtypes, in that they empty better because the longitudinal muscles may be intact. Now, did you say he asked if longitudinal muscles play a role in symptoms?
Yes.
And Ravi has demonstrated that a sustained contraction of the longitudinal muscles can sometimes be a correlate of chest pain, and the imbalance between cervical muscle and longitudinal muscle contraction can sometimes explain chest pain symptoms. Patients who have a pain predominant presentation within what we now termed the functional realm. And Ravi has demonstrated that an imbalance in the control mechanisms of the circular and longitudinal muscles function may partly explain that. So yes, longitudinal muscles are important. Don't identify the result of longitudinal contraction very well with our present manometry systems that look at pressure change in the esophageal lumen.
Okay, we have one final question for you, Prakash, and if you could keep it brief, we'll finish on time here. And this is related to the immune-mediated loss of nitrogen neurons. Do you see that more in the lower esophageal sphincter region or in the esophagus, or both in achalasia?
Excellent question. There are two pathways. One is where there is neuronal death. Everything goes. Type 1, type 2 achalasia, where there is no contraction in the esophageal body, abnormal LES relaxation. A second pathway where there is damage but not death, where you can have spotty loss of neuronal function. So you can have esophageal body, LES, both, either one more than the other. You get these spastic disorders, these incomplete patterns, EGJ outflow obstruction, hypercontractile with outflow obstruction. At least that's the hypothesis.
Okay, thanks. Well, I'd like to thank the panelists once again for these outstanding talks. If anybody wants to, you can contact the panelists directly and ask additional questions that you might have. I believe we got through all the questions that the audience had, so we probably won't be posting anything on Doc Matter, but this entire session will be available on YouTube within a day or so.